Now it started, and you can go ahead, Niklas.
Thank you, Anisha. Yes, Henrik said, this webinar series is then part of the Bridge Sweden, which is Vinnova financed project spanning over 3 years, which aims to help projects going faster from the discovery phase into the clinical phase, where we have different actors working both in the discovery part, myself from the drug discovery and development platform at SweLife Lab. We also have Stem Cell Center and pre-GMP facility at Lund University. And then moving towards the sort of clinical phase, we have the different innovation offices at KI, UU, and LU, and then also Stuns and Umeå Biotech Incubator helping out. But the main part of this bridge project is to facilitate the move from discovery to clinical development, and then we have the cell line development as sort of our key factor here. And this is a facility that is now up and running at the drug discovery and development at SweLife Lab. And then we wish to move from the discovery cell line development into then test a center. We can do some process development and scale up. And in this project, we also have 4 different academic groups that we're working together with that are very, very interesting. And one question that early sort of comes up is regarding regulatory elements. So I'm also looking forward to this webinar very much. But if you have any questions regarding the Bridge project at large or cell line development, you can contact me, Niklas Thalén, at the Drug Discovery and Development platform later. And I think that's it, Henrik, a very, very brief introduction to Bridge.
Yeah. Great. And I forgot to introduce myself. I was so excited by this. Erik Istvad, I'm project manager at Stuns Life Science. And like Niklas said, I mean, this is one of the pillar stones when it comes to the regulatory. So it's going to be super interesting to hear what Emma and Marie have to say. You're welcome to start your presentation. And we'll take the questions afterwards. You'll have your presentation for approximately 30 minutes, and then we have a 15-minute Q&A. So prepare any questions. You can give them directly, or you can write them in the chat, and I will bring them up as well. So feel free to just remember any questions that you have. So with that said, you're welcome to start your presentation.
Thank you. It's great to be here. And of course, regulatory is, I completely agree, it's a key pillar stone in the drug development. And RegSmart, just a few words about RegSmart. We are a consultancy firm based in Uppsala, working with drug development and regulatory strategies in drug development from early preclinical up to market. And we both have biotech and med tech in consultants and experts at RegSmart. And I'm Marie Gårdmark. I'm the CEO of RegSmart, and I have a background from as a regulator from the Swedish agency, but I've also been in drug industry working with global regulatory strategies for a number of years. And we also have Emma here.
Yes, Emma Eriksson, and I have worked with ATMP development in academia and small industry or biotech industry, and then spanning preclinical and clinical development, and also a lot of focus on the process development and quality of the product, which I will talk more about today.
Yes. So we will start. I will start with a couple of slides around very high level introduction to regulatory and regulatory strategies and what it's all about. And then Emma will talk more about the CMC questions and more dig a bit deeper into that as we go along. So let's see if we can switch slide here. Right. So regulatory. Thinking about regulatory is important, and starting early to think about regulatory. It's something that we know that investors are looking for this and understanding also the strategy all up to approval, up to markets, basically, because it's, even though very early on you might not have all the information available, but at least have a starting point with the end in mind is important also for the regulatory strategy. And the key thing to start with is the target product profile. Understanding what is the product going to look like when it's on the market, what patient population, what indication, what is the vision, and so forth. What are the main claims you want to do? Because that will also drive the regulatory strategy moving forward. So regulatory is very much linked to, of course, both science and the data that you get along the way, but also the commercial intention with the product. And what is really driving the regulatory pathway and the regulatory decisions? Well, of course, there are an interest also from the regulators in looking into the patient and the patient need. I guess that's what we all want to in the end. But then as a basis, we have the legislation and the regulation, of course, which looks a bit different in different areas and different regions of the world. But they tend to be fairly broad and not so detailed. But to give a bit more guidance, regulators are, of course, then developing guidelines. I will come back to that, what they really mean. But something that is also very important to look at is the precedents. What kind of decisions have been made before? How did the regulators evaluate a specific type of information previously? And there's a lot of information out there to look into. A lot of the information is published. Perhaps not so much when it comes to the CMC part, actually, but for other parts, preclinical and clinical parts, it's a lot of information available in the public domain. And in the end, it's the data that is driving the regulatory decisions in the end. So when we talk about guidelines, how important are the guidelines and must they be followed? And from what I've been working in this field for a long time, I always say that you should always be a bit, you should be aware of the guidelines, of course, but it's not a legislation, it's not a legal document, it's guidelines. And it's written from the regulators to give, this is the current thinking that we have and the current thinking when we wrote the guideline. But it's not the regulators that are driving the research. It's the companies that are driving the research and kind of moving the regulators eventually. So it's very, very important that as a company and a researcher that you look beyond the guidelines, because otherwise the agency's thinking will never develop. So it's a kind of an interplay between research and drug development and the regulators, which is important to be aware of. And in the end, it's always the benefit risk that is important. And if you see a huge benefit, huge unmet need, of course, you, as a regulator, are prepared to take a bit more risk as well. And also important to understand is that there will always be huge amount of uncertainty when you kind of search for, or when you are in the approval phase. There are just
finally
limited amount of data.
understand why E switched his position.
What do you mean?
You can light shine my eye.
Once we, you can always change if you want, but like when we, I
think it's
really
almost
Please
okay.
mute.
Sorry,
Yeah.
can you mute?
Yeah. So and just a few words about the regulatory systems in because what I just spoke about, that's kind of a common for all regulators in globally. But if we dig a bit more into the systems in Europe and US, they are a bit different, and I will not dig into the details here. But what is important to understand is that in Europe, of course, we do have a common regulation in Europe. We have so the legislation is similar, but it's the development is still you still develop your drug. You do your clinical studies, separate studies. There's no one in EU having a kind of an oversight over the development program. Whereas in US, if you start working with FDA, they will have this oversight over the program, which is both good and bad, of course, but it's fundamentally different. And in US, you do have a division that is responsible, an FDA division that is responsible for your indication area. Whereas in Europe, you have 27 countries that are all communicating, and they are all being part of the review process and the regulatory process. But it's kind of difficult to understand who is really making the decisions in Europe. It's just a different kind of system in the two regions, which is important to understand when you decide on your regulatory strategy. There are, of course, a toolbox that we can use to understand and to discuss the development program with the regulators. And an important piece of this toolbox is the scientific advice possibilities that we have in all regions. I mean, this is a good opportunity. This is a good thing, and it's very useful, especially when we are deviating from guidelines, when we are doing something completely new modalities, new targets, new technology, where we also might want to educate the authorities. Then scientific advice is a very good tool to use. And we also know that the people want investing in companies or in life science industry, they tend to want to see that the projects have been discussed with the regulators at some point. So it's a good toolbox. But is it always good to seek advice? Well, it really depends. Yeah, I mean, sometimes, well, it costs money, of course. It takes time. And regulators, they want to help, but sometimes they can be too helpful. You may get advice that you don't want. So you need to be prepared to get response that you don't really want to have. And it might end up in the meeting minutes, which you then have to keep with you during the rest of your development pathway. So not always good, but on the other hand, as you know, I said on the last slide, sometimes you really do want that early feedback, and you do want to understand how the regulators think about this specific new pathway or whatever. And if you're going in for a great or new, very huge investment, for example, moving into phase 3, yes, you do want to have the feedback from the regulators. So it's good and bad, always. And the regulators, they have also put together a number of initiatives to stimulate drug development, especially in where they see that there is a huge unmet need. And that's why there are different ways of increasing the communication with both the European and the US authorities through, for example, various types of designation, fast track designation procedures. There are also ways of accelerating the approval process in both regions. So there are this is also part of the regulatory toolbox to use when it's when there is when the project is in the right phase and with the right data. And I think I stopped there. And we move to you, Emma.
Yes. So I want to take some time and talk about a part of drug development. And focus then on the key CMC considerations for biological drug development. So the key question is, what makes biologics unique to other drugs? They are large and complex molecules that are produced in living cells and spans a lot of different types of modality or different types of drugs, from monoclonal antibodies and proteins and vaccines to the advanced therapy, medicine and products. And since they are produced in living cells, it's important to know that the product is influenced by the differences in the production process. So, for example, differences in temperature, pH, or shear stress during the production will affect or have an influence on how the product will look like in the end, which means that biologics have an intrinsic variability compared with small molecules, which means that two batches of the same product are never 100% identical with each other. So what I really want to emphasize here is that the process is part of the product, and this will lead to that we will have regulatory expectations on a strong process understanding. So if you don't know what CMC stands for, it's the abbreviation for chemistry, manufacturing, and control. And it aims to answer core questions related to what is the product, how is it made, and how do we ensure the quality over time. And the reason for this is so that we can develop a process that is scalable, reproducible, and comply with quality standards to ensure safety and efficacy of the product between different batches. And in the end, when moving towards market approval, we should have a well-described process that is justified and validated, which also includes all the analytical methods and assays that are used to control it. And CMC is not something you do in the beginning of drug development. It will run through the entire development phase from the preclinical to the commercial stages, as we learn more about the product and define our process better and validate the different steps. The CMC has different building blocks that we need to develop and understand. And the first one is the understanding of the product. We have to know the structure, for example, the immunological properties, the biological activity of our drug, but also things related to purity and impurities that comes from the process or the product, and different contaminants that can be introduced into the production, which, for example, can be of viral or bacterial origin. We have to have a good understanding and develop our process, how it will look like, as we heard on the last bridge seminar, the cell line creation, but also then moving into the upstream production in cells, the downstream purification of our product, and the viral inactivation and removal, and lastly, the formulation of the drug and how that will look like. We have to have a well-developed control strategy with analytical testing for releasing of the batches. but also different in process controls to ensure the consistency and quality of our drug product. We also have to think about and show stability so that we can ensure that the product is functional over time. And lastly, we also have to think about how this will look like in the end, not just during the development phase, but moving into the manufacturing at a GMP level, and also later the supply of the drug, both for then clinical and commercial batches. So I want to take some minutes to focus on 3 crucial points to biological drug development. And that's the potency assay development, the immunogenicity of the drug, and comparability studies, all that have a huge part of the regulatory of the CMC regulatory. So potency assays, if you are not familiar with the terms, it's the quantitative measurement of biologic activity of a product that should reflect the mechanism of actions in humans. And it's very important that this assay can detect differences in the quality of the product, since it plays a part both in the release of different batches for use in humans, but also during it is utilized during stability programs. I want to give some examples of what it means that it should be relevant for the mechanism of action. So if we have a look at monoclonal antibodies, we can have antibodies where the mechanism of action is that the antibody should be able to bind and neutralize a target, which means that we can have a potency assay that shows that the antibody actually binds to the target. So just an in vitro binding assay. But for example, if our antibody has an effector function in the form of that it has ADCC or CDCC which is relevant for the clinical activity of the drug, then we might need to utilize a cell-based bioassay where we show that this actually happens. And to have a potency assay is a regulatory requirement. The potency assay has to be fully validated before entering pivotal studies, which is important to remember. So it's good to start early with the development of the potency assays, and they don't have to be one single assay to start with. You can utilize several, and then as you move the program forward, decide which one is most relevant for your product. I also want to talk a bit about immunogenicity. So that's the formation of an immune response against a product, which, for example, can be antibodies that have or do not have neutralizing capacity in humans. And this can have an impact on the efficacy and safety of the drug. It's something that's investigated during clinical development in the form that you have to measure anti-drug antibodies. And the reason for why I want to mention it here is that it's, as we can understand, it is connected to the sequence or structure of our active substance. But why I really want to mention it is that it also has a component where it can be process related, so related to the manufacturing of the drug, in the form of if you have impurities in the form of aggregates, those can lead to generation of anti-drug antibodies. And lastly, I also want to take some time to talk about comparability studies. So comparability, so when you move through the development of the drug, you might find yourself in a situation where you have to do changes to the manufacturing process. This can, for example, be that you have to improve the process. You have to change the scale, which will lead to differences in the manufacturing. Improving the stability by formulation changes, or new or different regulatory requirements that your manufacturing process has to comply to. So comparability is where you then have to provide evidence that the changes that you are making will not have an adverse impact on quality, safety, and efficacy of the drug product. And you don't have to show that that the pre and post change is identical. You have to show that they are highly similar. And this can be done in the best case scenario. You have to do it through just show it by analytical testing of the pre and post change product. But it can also be that the changes that you have introduced need to be shown that they are comparable in preclinical or clinical studies with different bridging studies. And comparability studies is something one should not be scared of, but something that you should plan that it can happen as you move through the development. So it's always important to remember to have a lot of samples saved so that one can compare the pre and post changes without having to produce additional batches that are to then the pre-change manufacturing process. So just a quick summary. Biologics require control of complex living systems. They are an early product and process understanding will save in the end save time and risk. And robust analytical and control strategies are critical. And that was the end of our presentation.
Perfect. Thank you very much. Super interesting. Some really good information here. And now the questions are rolling in here. I have a question from Olala. Do you want to ask it yourself or should I read it up? I'll read it. Is CMC strictly for biologics or does it extend to biomedical devices, implantables, for example?
You're thinking if your drug is a combination between a medical device and a biological drug.
Yeah, or is it, maybe you want to elaborate this one, or I think is it just that CMC, is it strictly for biologics or does it apply to any other field?
The CMC is for all pharmaceutical drug development, so regardless of it's a biologic drug or if it's a small molecule, they all go through the CMC part, and it's an essential part of the documentation that you put in when you go for a clinical trial or market authorization. So it's the module 3 of the what it has to be.
And for medical device, there's a completely different regulation.
Okay.
Yes.
But this was an example of dispensing implant. That would be on the medtech device side then.
Yeah, I guess if there's but that's if you think about again a combination product where you have a specific piece that is biologic or a small substance, it's a CMC. I mean, you still need to do have a control over the that part of the dossier as well.
Yeah. And a small follow-up question: what happens when comparability study indicates significant improved efficiency?
Yeah, that's a really interesting question, depending on how. So I guess you mean that in a bridging clinical trial, you show that you have improved efficiency of the drug.
Yeah, I mean, then it could be a very good situation, of course, but I guess then you have to do your studies to confirm that improved efficacy in phase 3 trial. I guess it depends where you have this increased comparability efficacy. If it's I mean, it could be a preclinical model, I guess. And then of course you need to confirm it in the clinical setting. So it's it could be a good thing, but it could also be a costly thing. It depends on how good it is, if it's a commercially a strong thing or not.
Yeah. Great. We got 2 questions here from Georgia. And I will start off with the first one. How can you effectively align FDA and EMA regulatory requirements to create a harmonized global regulatory strategy for biologic products and process development?
Oh, it's a good question.
And a tough one, I guess.
Yeah, but I think that's really what the regulatory strategy is, understanding, is there sometimes that that the difference is not that huge, so your program will fulfill the requirements in the big regions. But in other cases, you know that there are differences in the way that the regulators are thinking, or maybe you just don't know because it's something new. So then you need to discuss, make sure that you have the right consultations at the right time with both FDA and relevant EU authorities. And again, also consider what's your main market. So far, US has been the main market for many of at least our clients, and so kind of it's been more important what FDA said than the EU regulators, to be honest. But again, it depends on and we don't know about the future. Everything's happening on the other side of the Atlantic. But yeah.
Yeah, so I refer also to the fact that the FDA it's more based on risk-based control of, for example, of the raw materials, and the EMA is not. So it's more based on, I would say, more GMP compliant of the materials. So this was my thinking.
Yeah. Comment on that?
Yeah, I would say.
So it's 2 different strategies.
Yes, but still, if you want to move ahead with both of them at the same time and go, then you really have to think about who will have the strongest requirements, because then you will get the other one on the side.
Okay.
Because then you have to focus on if it will be a problem, and you have to go for then it's better to develop it as to the strongest requirements.
If you want to go.
If you want to go for both of them.
Yeah. But I mean, you're absolutely right in saying that on a generalizing, FDA tends to be a bit more approving on maybe wrong to say higher risk, but we've seen a number of approvals during the last couple of years where FDA said yes and EU said no. So and because they are sometimes interpreting the risk benefit or the benefit risk profile a bit different. So FDA might think that, well, we're not completely sure about the benefit here, but we still think it's sufficiently good. Whereas EMA said, well, we're not completely sure about the benefit here, so we will not approve. It's different ways of kind of going into the benefit risk profile and the benefit risk evaluation. So we've seen that for a number of products during the last couple of years.
Yes, and I think that FDA also has a real focus. If it is a clear unmet need, then that will also try.
Yes,
they will really go for it with the unmet need, because there you have. If there is no other drugs on the market, you know that these patients will need something, and even though in the end that the EMA says the efficacy of the drug is not that high, then there is something on the market for that patient population.
Yeah, and more leaving it to the prescriber.
Yeah, to and the insurance companies.
Yes, in the US.
Okay.
Okay, thank you.
Thank you. We have a question from Hamid. Do you want to ask it yourself or should I continue?
I also have another question to Emma.
Yeah.
Yeah, so
Go
how
on.
can you strengthen product understanding around, for example, potency assay, identity and purity to guide the process development and ensure that manufacturing changes do not alter the quality, safety, and efficacy of biological products?
I'm not sure if I heard the first you said. It was a little bit.
How can you strengthen the product understanding around the potency assay, identity, and purity?
I mean, it's just important to know that you have to focus on these parts and to start the development early of the potency assays, which I think if you look at some where there might be a lack is that you have to really see that the potency assay reflects the mechanism of action that is intended. I mean, what you really are saying, this is what happened inside the patients when you give the drug, and to see that you can develop or have an assay that reflects that. But it's also important to know that you are not limited to one assay. You might need to have several assays that shows that capitulates the entire picture. And just to have it in mind that it is important early to focus on the CMC development, because the earlier we start, the more knowledge and time we will have to actually have the time to focus on that. And if you have well-developed methods that you find that can really show the identity of your product, it can show the potency, it becomes so much easier when you have to do a comparability study, because then you have all these methods that can actually show all of and a lot of for the impurities in form of aggregates and difference. I mean, they are also part of the pharmacopoeia, so you can also you also utilize that you have these methods. I hope that.
Yes.
Yeah. I'll continue with Hamid's question. Can guidelines be internally adjusted to align with specific processes, local regulations, manufacturing considerations, provided there is appropriate justifications? You touched a bit on that, but would you elaborate?
Well, I think, guidelines, as I said, are only guidelines. As long as there is data and a good rationale, you can deviate from guidelines, and sometimes there are no guidelines for specific areas. So you just have to invent the pathway yourself. But it's important that you can link the data to a rationale and to the specific claims that you want to do, or a specific process or whatever, and present it in a way to the regulators so that they understand the risks, or because that's what it's all about. It's for the regulators to understand what is the risk with this with this compound, with this process in relation to the specific benefit or claim you're making. I don't know if that answered your question.
See if Hamid is... Yes, perfect. And we get a follow-up questions here from Alola. A side note, what kind of qualifications are needed to switch careers from experimental biology to join you in regulatory affairs? That was an interesting question.
That's a good question. Well, Yeah, I mean, it's yeah. It's I mean, the regulator is not something you really. There's no education per se. I mean, there are nowadays there are specific courses at the university that are focusing a bit on regulatory from different angles, and of course also there are courses at, for example, Läkemedelsakademin, focusing on regulatory aspects in in CMC and regulatory strategies and many different ways. And then it's a lot of on-the-job training, I think. It's really there are possibilities to learn and then working with different projects and to me regulatory is very much common sense. It's understanding the data, understanding the science. It's important, but also understanding what is the intention of the regulators here, and reading between the lines. What is the what is the point, or what is the really the really the intention of the regulation and the guidelines. And it's a lot of common sense, I would say.
Great. We got one final question here from Albert. I mean, we're 14 minutes or 12 here, so but I will see if you can answer this briefly. Can you comment on biosimilars and difference regarding potency, assay, immunogenicity, and comparability studies? I guess it's a complicated question for you guys, but
yeah, it's a huge question.
yeah, we're running out of time, but for those who need to leave, you can leave. For others, you can hang on and listen to get a clear answer to this one.
Yeah. I mean, I'm not. Yeah, it's been a while since I worked with biosimilars, but it's really nowadays. I have really forgotten the exact wording of the questions, but many times you do need to do clinical studies in order to prove that you have to prove the biosimilarity, which is of course very much different from the generics, where you can prove similarity sometimes just from in vitro data and sometimes by equivalent studies. Can you phrase the question again?
Yeah, or maybe Albert, if you want to join in and ask the question yourself. I'll take it. Can you comment on biosimilars and the difference regarding potency, assay, immunogenicity, and comparability studies?
Yeah. So it tends to be more from a it's because it's, I mean, the comparability, of course, you need to show in some way, either usually then clinically, but sometimes it's sufficient with non-clinical or even if you can characterize your compound or your in a sufficient way. But the immunogenicity, I mean, I guess it's the same thing. I don't think I'm really equipped to respond to that question without doing some digging. But from a comparability perspective, a biosimilar immunogenicity. I mean, if you do the clinical studies, you will look at the immunogenicity in the clinical studies, of course. But can you do that also in the from a CMC perspective, just in the characterization of the compound?
I'm not sure, actually. I would have to have a look.
Yeah,
but I think that I mean, immunology, I mean, how it looks like. You really have to. I think you have to show it in the clinic, also.
Well, you can hear that we're not completely sure here.
Not sure about the
we'll need to dig deeper into that question. But as always, I mean, if you have any questions regarding regulatory, you're welcome to contact Emma and Marie, you got their contact information. I'll send it out. We'll also send the recording out to you guys so you can follow this up. And if you have any questions regarding the bridge project, feel free to contact me or Niklas or Anisha, and we'll help you out. And with that said, I think we will round this webinar off. Super thanks for having us here and giving your introduction to regulatory affairs. It was really interesting, and I think it was super, important for us to get this kind of knowledge. So big thanks for you at RegSmart for joining us today, and for all of you who participated. So, and I just want to finish off with saying that the fun is not over. At the 22nd of April, we'll talk about preclinical studies. So give that in your calendar, mark that as 11:30 to 12:15, and we'll send out some information and about that in the near future here. So, great. Thank you, guys.
Thank you.
Thank you.
Bye-bye.
Bye-bye.